Hello and welcome dear students. Today we are going to discuss about the topic Introduction to Epigenetics. The main objectives of today's lecture are to know the basics of epigenetics, to know different terminology used in epigenetics, to discuss the molecular basis of epigenetics, to discuss how the repair of oxidative DNA damage can alter epigenetic markers, to discuss the techniques used to study epigenetics, to discuss about chromatin immunoprecipitation, and finally to discuss about the methylation sensitive restriction enzymes. Dear students, let's start with the introduction first. In biology, epigenetics is the study of stable phenotypic changes known as marks that do not involve alteration in the DNA sequence. Epigenetics most often involves changes that affect the regulation of gene expression, but the term can also be used to describe any heritable phenotypic change. Such effects on cellular and physiological phenotype traits may result from external or environmental factors or be part of normal development. The term also refers to the mechanism of change, functionally relevant alterations to the genome that do not involve mutation of the nucleotide sequence. Examples of mechanisms that produce such change are DNA methylation and histone modification, each of which alters how genes are expressed without altering the underlying DNA sequence. Gene expression can be controlled through the action of repressor proteins that attach to silencer regions of the DNA. These epigenetic changes may last through cell divisions for the duration of the cell's life and may also last for multiple generations even though they do not involve change in the underlying DNA sequence of the organism. Instead, non-genetic factors cause the organism's genes to behave or express themselves differently. One example of an epigenetic change in eukaryotic biology is the process of cellular differentiation. During morphogenesis, totipotent stem cells become the various pluripotent cell lines of the embryo, which in turn become fully differentiated cells. In other words, as a single fertilized egg cell, the zygote continues to divide, the resulting daughter cells change into all the different cell types in an organism, including neurons, muscle cells, epithelium, endothelium or blood vessels, etc., by activating some genes while inhibiting the expression of others. Dear students, let's now discuss about the different terminology used in epigenetics. The term epigenetics in its contemporary usage emerged in the 1990s, but for some years has been used with somewhat variable meanings. A definition of the concept of epigenetic trait as a stably heritable phenotype resulting from change in a chromosome without alterations in the DNA sequence was formulated in 2008, although alternate definitions that include non-heritable traits are still being used widely. Dear students, let's now discuss about the Weddington's canalization of 1940s. The hypothesis of epigenetic change affecting the expression of chromosome was put forth by the Russian biologist Nikolai Koltsov. From the genetic meaning and the associated adjective epigenetic, British embryologist C. H. Weddington coined term epigenetics in 1942 as pertaining to epigenesis in parallel to Valentin Hecker's phenogenetics. Epigenesis in the context of the biology of that period referred to the differentiation of cells from their initial totipotent state during embryonic development. When Weddington coined the term, the physical nature of genes and their role in heredity was not known. He used it instead as a conceptual model of how genetic components might interact with their surroundings to produce a phenotype. He used the phrase epigenetic landscape as a metaphor for biological development. In recent times, Waddington's notion of the epigenetic landscape has been rigorously formalized in the context of the systems dynamics state approach to the study of cell fate. Dear students, let's now discuss the contemporary definitions of epigenetics. Robin Holliday defined in 1990 epigenetics as the study of the mechanism of temporal and spatial control of gene activity during the development of complex organisms. Thus, in its broadest sense, epigenetics can be used to describe anything other than DNA sequence that influences the development of an organism. More recent usage of the word in biology follows stricter definitions. As defined by Arthur Riggs and colleagues, it is the study of mitotically and or meiotically heritable change in gene function that cannot be explained by change in DNA sequence. The term has also been used, however, to describe processes which have not been demonstrated to be heritable, such as some forms of histone modification. Consequently, there are attempts to redefine epigenetics in broader terms that would avoid the constraints of requiring heritability. For example, Adrian Bird defined epigenetics as the structural adaptation of chromosomal regions so as to register, signal, or perpetuate altered activity states. This definition would be inclusive of transient modifications associated with DNA repair or cell cycle phases, as well as stable change maintained across multiple cell generations, but exclude others such as templating of membrane architecture and prions unless they impinge on chromosome function. Such redefinitions, however, are not universally accepted and are still subject to debate. In 2008, a consensus definition of the epigenetic trait, a stable, heritable phenotype resulting from change in a chromosome without alteration in the DNA sequence, was put forward. The similarity of the word to genetics has generated many parallel usage. The epigenome is a parallel to the word genome, referring to the overall epigenetic state of a cell. And epigenomics refers to global analysis of epigenetic change across the entire genome. The phrase genetic code has also been adapted. The epigenetic code has been used to describe the set of epigenetic features that create different phenotypes in different cells from the same underlying DNA sequence. Taken to its extreme, the epigenetic code could represent the total state of the cell with the position of each molecule accounted for in an epigenomic map, a diagrammatic representation of the gene expression. DNA methylation and histone modification stats of a particular genomic region. More typically, the term is used in reference to systematic efforts to measure specific relevant forms of epigenetic information such as the histone code or DNA methylation patterns. Dear students, let's discuss the epigenetics in terms of the developmental psychology. In a sense somewhat unrelated to its use in any biological disciplines, the term epigenetic has also been used in developmental psychology to describe psychological development as the result of an ongoing bidirectional interchange between heredity and the environment. Interactive ideas of development have been discussed in various forms and under various names throughout the 19th and 20th centuries. An early version was proposed among the founding statements in embryology by Karl Ernst von Baer and popularized by Ernest Haeckel. A radical epigenetic view known as physiological epigenesis was developed by Paul Winterburt. Another variation, probabilistic epigenesis, was presented by Gilbert Gottlieb in 2003. This view encompasses all the possible developing factors on an organism and how they not only influence the organism and each other, but how the organism also influences its own development. Gottlieb gave an example of rhesus monkeys where infants that did not receive typical maternal care lacked serotonin, which in turn made them more aggressive as they got older. On another note, the long-standing notion cells that fire together wire together derives from Hebbian theory, which asserts that synaptogenesis, a developmental process with great epigenetic procedure, depends on the activity of the respective synapses within a neural network. Where experience alters the excitability of neurons, increased neural activity has been linked to increased demethylation. The developmental psychologist Erikson wrote of an epigenetic principle in his 1968 book Identity, Youth and Crisis, encompassing the notion that we develop through an unfolding of our personality in predeterminal states and that our environment and surrounding culture influence how we progress through these stages. This biological unfolding in relation to our socio-cultural settings is done in stages of psychological development, where progress through each stage is in part determined by our success or lack of success in all the previous stages. Although empirical studies have yielded discrepant results, epigenetic modifications are thought to be a biological mechanism for transgenerational trauma. Dear students, let's now discuss about the molecular basis of epigenetics. Epigenetic changes modify the activation of certain genes but not the genetic code or the sequence of the DNA. The microstructure, not code of DNA itself or the associated chromatin proteins, may be modified, causing activation or silencing of that particular gene. This mechanism enables differentiated cells in a multicellular organism to express only the genes that are necessary for their own activity. Epigenetic genes are preserved when cells divide. Most epigenetic changes only occur within the course of one individual organism's lifetime. However, these epigenetic changes can be transmitted to the organism's offsprings through a process called transgenerational epigenetic inheritance. Moreover, if gene inactivation occurs in a sperm or egg cell that results in fertilization, this epigenetic modification may also be transferred to the next generation. Specific epigenetic processes include paramutation, bookmarking, imprinting, gene silencing, position effect, transfection, maternal effects, X chromosome inactivation, DNA methylation, reprogramming, the progress of carcinogenesis, many effects of teratogens, regulation of histone modifications, and heterochromatin, and technical limitations affecting parthenogenesis and cloning. Dear students, let's discuss the role of DNA damage. DNA damage can also cause epigenetic changes. DNA damage is very frequent, occurring on average about 60,000 times a day per cell of the human body. These damages are largely repaired. However, epigenetic changes can still remain at the site of DNA repair. In particular, a double-strand break in DNA can initiate unprogrammed epigenetic gene silencing, both by causing DNA methylation as well as by promoting silencing types of histone modifications, that is chromatin remodeling. In addition, the enzyme PARP1 poly-ADP ribose polymerase and its product poly-ADP ribose PAR accumulate at sites of DNA damage as part of the repair process. This accumulation in turn directs recruitment and activation of the chromatin remodeling protein ALC1 that can cause nucleosome remodeling. Nucleosome remodeling has been found to cause, for instance, epigenetic silencing of DNA repair gene MLH1. DNA damaging chemicals such as benzene, hydroquinone, styrene, carbon tetrachloride, and trichloroethylene cause considerable hypomethylation of DNA, some through the activation of oxidative stress pathways. Foods are known to alter the epigenetics of rats on different diets. Some food components epigenetically increase the level of DNA repair enzymes such as MGMT and MLH1 and P53. Other food components can reduce DNA damage such as soy isoflavones. In one study, markers for oxidative stress such as modified nucleotides that can result from DNA damage were decreased by a 3-week diet supplemented with soy. A decrease in oxidative DNA damage was also observed 2 hours after consumption of anthocyanin-rich bilberry or pomace extract. Dear students, let's now discuss the role of DNA repair. Damage to DNA is very common and is constantly being repaired. Epigenetic alterations can accompany DNA repair of oxidative damage or double-stranded breaks. In human cells, oxidative DNA damage occurs about 10,000 times a day, and DNA double-strand breaks occur about 10 to 50 times a cell cycle in somatic replicating cells. The selective advantage of DNA repair is to allow the cell to survive in the face of DNA damage. The selective advantage of epigenetic alterations that occur with DNA repair is not clear. Dear students, repair of oxidative DNA damage can alter epigenetic markers. In the steady state, with endogenous damage occurring and being repaired, there are about 2,400 oxidative damaged guanines that form 8-oxo-2-deoxyguanosine in the average mammalian cell DNA. 8-OHDG constitutes about 5% of the oxidative damage commonly present in DNA. The oxidized guanines do not occur randomly among all guanines in DNA. There is a sequence preference for the guanine at a methylated CpG site, a cysteine followed by guanine along its 5-3 direction, and where the cytosine is methylated 5-MCpG, a 5-MCpG site has the lowest ionization potential for guanine oxidation. Oxidized guanine has mispairing potential and is mutagenic. Oxyguanine glycolase is the primary enzyme responsible for the accession of the oxidized guanine during DNA repair. OGG1 finds and binds to an 8-OHDG within a few seconds. However, OGG1 does not immediately excise 8-OHDG. As an example, when human mammary epithelial cells were treated with H2O2, that is hydrogen peroxide, for 6 hours, OHDG increased about 3.5-fold in DNA, and this caused about 80% demethylation of the 5-methyl cytosines in the genome. So demethylation of CpGs in a gene promoter by TET enzyme activity increases transcription of the gene into messenger RNA. In cells treated with H2O2, that is hydrogen peroxide, one particular gene was examined, BACE1. The methylation level of the BACE1 CpG island was reduced, an epigenetic alteration, and this allowed about 6.5-fold increase of expression of BACE1 messenger RNA. While 6 hours incubation with the hydrogen peroxide caused considerable demethylation of the 5-MCpG sites, shorter times of hydrogen peroxide incubation appear to promote other epigenetic alterations. So treatment of cells with hydrogen peroxide for 30 minutes causes the mismatch repair protein heterodimer MSH2, MSH6 to recruit DNA methyltransferase 1 to sites of some kinds of oxidative DNA damage. This could cause increased methylation of cytosines at these locations. Dear students, let's discuss how the homologous recombinational repair alters epigenetic markers. During homologous recombinational repair of the double-strand break, the involvement of DNMT1 causes the two repaired strands of DNA to have different levels of methylated cytosines. One strand becomes frequently methylated at about 21 CpG sites downstream of the repair double-strand break. The other DNA strand loses methylation at about 6 CpG sites that were previously methylated downstream of the double-strand break, as well as losing methylation at about 5 CpG sites that were previously methylated upstream of the double-strand break. When the chromosome is replicated, this gives rise to one daughter chromosome that is heavily methylated downstream of the previous break site and one that is unmethylated in the region both upstream and downstream of the previous break site. With respect to the gene that was broken by the double-strand break, half of the progeny cells express that gene at a high level, and in the other half of the progeny cells, expression of that gene is repressed. When clones of these cells were maintained for 3 years, the new methylation patterns were maintained over that time period. In mice with a CRISPR-mediated homology-directed recombination insertion in their genome, there are a large number of increased methylations of CpG sites within the double-strand break-associated insertion. Dear students, let's talk about how the non-homologous end joining can cause some epigenetic marker alterations. Non-homologous end joining repair of a double-stranded break can cause a small number of demethylations of pre-existing cytosine DNA methylations downstream of the repair double-stranded break. Further, it has been evaluated that NHEJ of a DNA double-stranded break in a cell could give rise to some progeny cells having repressed expression of the gene harboring the initial double-stranded break, and some progeny having high expression of that gene due to epigenetic alteration associated with NHEJ repair. The frequency of epigenetic alterations causing repression of a gene after an NHEJ repair of a DNA double-strand break in that gene may be about 0.9%. Dear students, let's now discuss about the techniques used to study the epigenetics. Epigenetic research uses a wide range of molecular biological techniques to further understand of epigenetic phenomena. These techniques include chromatin immunoprecipitation together with its large-scale variants that's like CHLP on-chip and CHLP sequence, fluorescent in situ hybridization, methylation sensitive restriction enzymes, DNA adenine methyltransferase identification, and bisulfite sequencing. Furthermore, the use of bioinformatics methods has a role in computational epigenetics. Dear students, let's first discuss the chromatin immunoprecipitation. Chromatin immunoprecipitation has helped bridge the gap between DNA and epigenetic interactions. With the use of CHLP, researchers are able to make findings in regards to gene regulation, transcription mechanisms, and chromatin structure. The second one is fluorescent in situ hybridization. Fluorescent in situ hybridization, or FISH, is very important to understand epigenetic mechanisms. FISH can be used to find the location of genes on chromosomes as well as finding non-coding RNAs. FISH is predominantly used for detecting chromosomal abnormalities in humans. Another one is methylation sensitive restriction enzymes. Methylation sensitive restriction enzymes paired with PCR, that is the polymerase chain reaction, is a way to evaluate methylation in DNA, specifically the CpG sites. If DNA is methylated, the restriction enzymes will not cleave the strand. Contrarily, if the DNA is not methylated, the enzymes will cleave the strand and it will be amplified by PCR. Another technique is bisulfite sequencing. Bisulfite sequencing is another way to evaluate DNA methylation. Cytosine will be changed to uracil from being treated with sodium bisulfite, whereas methylated cytosines will not be affected. Dear students, let's now conclude the lecture. Epigenetics is the study of how cells control gene activity without changing the DNA sequence. Epigenetic changes are modifications to DNA that regulate whether genes are turned on or turned off. These modifications are attached to DNA and do not change the sequence of the DNA building blocks, that is the nucleotides. Within the complete set of DNA in a cell, that is the genome, all of the modifications that regulate the activity, that is the expression of the genes, is known as epigenome. Because epigenetic changes help determine whether genes are turned on or off, they influence the production of proteins in cells. This regulation helps to ensure that each cell produces only proteins that are necessary for its function. For example, proteins that promote bone growth are not produced in muscle cells. Patterns of epigenetic modification vary among individuals in different tissues within an individual and even in different cells within a tissue. Environmental influences such as the person's diet and exposure to pollutants can impact the epigenome. Epigenetic modifications can be maintained from cell to cell as cells divide, and in some cases can be inherited through the generations. A common type of epigenetic modification is called DNA methylation. DNA methylation involves the attachment of small chemical groups called methyl groups, each consisting of one carbon atom and 3 hydrogen atoms, to DNA building blocks. When methyl groups are present on a gene, that gene is turned off or silenced, and no protein is produced from that gene. Another common epigenetic change is histone modification. Histones are structural proteins in the cell nucleus. DNA wraps around histones, giving chromosomes their shape. Histones can be modified by the addition or removal of chemical groups such as methyl groups or acetyl groups, each consisting of 2 carbon, 3 hydrogen, and 1 oxygen atoms. The chemical groups influence how tightly the DNA is wrapped around histones, which affect whether a gene can be turned on or off. Errors in the epigenetic process, such as modification of the wrong gene or failure to add a chemical group to a particular gene or histone, can lead to abnormal gene activity or inactivity. Altered gene activity, including that caused by epigenetic errors, is a common cause of genetic disorders. So conditions such as cancers, metabolic disorders, and degenerative disorders have been found to be related to epigenetic errors. Scientists continue to explore the relationship between the genome and the chemical compounds that modify it. In particular, they are studying the effects that epigenetic modifications and errors have on gene function, protein production, and human health. Dear students, it was all about today's lecture regarding introduction of epigenetics. Hope you have understood it well. See you next time with a new topic. Till then, take care and goodbye.